Okay, let's get started with today's session. So today we are discussing the final week, week 8, where we covered higher dimensional data. So and this will be the last session. And by the way, there is an announcement. So tomorrow we have an extra session. It will be like a revision session from 6 p.m. to 8 p.m. And the link will be the same. They have already sent the email. I hope you all received it. So we will just revise all the sessions from week 1 up to week 8 revision session. So that will be, yeah, tomorrow from 6 p.m. to 8 p.m. Please note the timing. It starts at 6 p.m. itself. And after this, directly after today's class and tomorrow's revision class, you will be having your exam next week, I think. Okay, so let's go ahead with the session. So I can go to full screen. Yeah. So in biology, there are some experiments where we get like huge quantities of data. So for example, one example is single cell RNA sequencing. So you will have multiple cells, like thousands of cells, and you will be looking at multiple genes. So you should know how to handle, how to visualize, how to analyze this kind of complicated data. And another example is high throughput microscopy. For example, you may have a 96 well plate, and from each well, you may take maybe even hundreds of images. So, to handle these kind of big types of data, we do something called dimensionality reduction. So, dimensionality reduction means so typically we plot a graph of X versus Y. But when you do these kind of experiments, then we will be having multiple variables. So in dimensionality reduction, essentially we take out the important parts of the data and we just plot it. So for example, if you're doing RNA-seq and you're analyzing maybe hundreds of genes, but out of the hundreds, only some of them would be the actual important genes that are downregulated and upregulated. So in dimensionality reduction, we will identify that important part of the data and plot it and analyze it. So two of the most common methods of dimension reduction in biology are PCA, which is principal component analysis, and TISNI. So PCA, like I said, it stands for principal component analysis. So it projects like higher dimensional, maybe two, three into lower dimensional, like 2 dimensions or 3 dimensions. So higher dimensional data, it can be like maybe 10 variables, 20 variables, like that. I can explain with an example. I'll just get my pen. So one simple example would be uh, maybe your, okay, my pen is not working. Actually, just give me a couple of minutes. I'll just get my pen to work and then we can continue. Mm. Meanwhile, do you have any doubts from the lectures? Just trying to connect the pen. It's not yet connected. Mm. Let me reshare the screen now. Yeah, so typically, if you have just two variables, we can just plot an X versus Y plot. Maybe it will look something like this. But in your data, if it has maybe like gene 1 expression, gene 2 expression, and we have hundreds of cells. Okay. Is my screen still visible? I got disconnected, I think. Yeah, it's still sharing. Okay, I'll just continue then. Then if you have this kind of large quantity of data, what we do is we try to find out of these, we try to find out the principal component. So we call it PC1 or principal component 1 and PC2 or principal components 2. And then we plot the data based on these principal components, which has the important information from here. So usually this PC1 is like it has more weight, or it's more the data is more dependent on PC1 than PC2. So it's not necessary that you have just PC1 and PC2. You can also have a third PC3 in some cases or more. So essentially, we have to first find these principal components of the data. And to find the principal components, we have to make the covariance matrix based on the um, data that is the data that we have. So we have to first find the covariance matrix. And from that, we have to find the eigenvector and eigenvalue. So I hope all of you remember the definition of eigenvector and eigenvalue. So it's essentially A V bar is equal to lambda V bar. And here this uh, lambda is the eigenvalue. And once you have the eigenvalues and the eigenvectors, then you can project the data on these to get finally you'll get the principal component plot. So this is the first important thing. I think somebody has raised their hand. Uh, okay, so for the examination, what types of questions we can expect? So for the exam, you'll be having MCQ type of questions. So we'll be discussing some problems today, and we have been discussing some problems before as well. And like we already announced in the classes, you won't be getting any R coding, but there will be questions where an R code may be given and you have to find out what is the output, like questions like this. So directly you won't be asked to write the code, but maybe we can give one line of R code and you have to say what it does. And there will also be other type of questions based on the concepts. So you'll have questions from the coding part, like MCQ questions only, and you'll also have questions from the concepts part. Uh, yeah, so the exam pattern is MCQ based. And tomorrow we have a revision session. So we can go through more example problems in the session for tomorrow. Any other questions before we move on to the next concept? I'll just wait for a few seconds and then we can go to the next concept. So the next technique is TSNE, like we call it TSNE, but it's TSNE. It stands for T-distributed stochastic neighbor embedding. So I hope all of you may have come across these TSNE plots, especially with gene expression data sets. I can draw a sample for you. So typically, if you see, it looks like there are different clusters with different colors, and each color corresponds to like a related set of data points. Maybe there are a cluster of points like this. So this is also used when you have higher dimensional data, and it works on the probabilistic measure of distance and dissimilarity. So it looks something like this. I'm just drawing very roughly. And what you do in this case essentially is that you have to, again, from a higher dimension, the information or the, uh, yeah, essentially the information from the higher dimension is preserved, and you bring it to a lower dimension for easier visualization. So this is the main concept behind the t-SNE plot. And then, yeah, so this was the first lecture, I think. And then we had a lecture on doing PCA in R, and then t-SNE, and then we also have some codes for that. And the final concept that was discussed last week was diffusion maps for dimension reduction. And this is also again another technique for like big quantity data. So for diffusion maps, as the name suggests, it has to do with the process of random walks with the data. So here there are two important concepts. One is the concept called transition matrix, and the other is the concept called as diffusion distance. So in this diffusion maps for dimension reductions, we perform a random walk on the data. So that so to get the probability of going from one point to another, we draw this transition matrix between the different data points. So this is an important concept. And the second important concept in this diffusion maps is called the diffusion distance. So here, the diffusion distance, it essentially says how similar the 2 points are with respect to all the other points of the data set. So the good thing about this is that it preserves the connection between the points, because when we perform a random walk, we will be considering the connections between the different points as well. And it is also more robust to noise. So we have finished with the discussion on revision of the concept. So now we can solve some problems. So this is the first question. You can take a couple of minutes, look at the question and understand it, and you can type the answer in the chat box. Then we can discuss the solution. Okay, somebody has raised their hand again. Yes. Is my screen visible? Somebody has written that my screen is not visible. So I think for some of you it is not visible. Can you try to sign out and then sign in again? So once you have the answer, you can type it in the chat box. Then we can discuss the answer, the solution. Okay, so it's a fairly simple question. So let's discuss the answer now. So the question is based on this R function called PRCOM. And they are asking what is the purpose of the argument scale is equal to true. So, like from the name, you can guess that this setting scale equal to true will scale the different variables. Um, but for which of these will it scale such that the mean is one or variable becomes one? So typically for a PCA, we standardize the data before doing the analysis, especially when you look at, for example, gene expression data. Depending on the gene, there are wide ranges of gene expression data. So first we have to scale it. And in this PRCOM, the scale function will scale the all the variables such that their variance becomes one. So the correct answer is option C. So this is a very important function because it's used to perform PCA. So make sure that you know what is the function and what are the different inputs to the function. We'll also be discussing this in the R coding session. So we'll come back to this again. So let's go to the second question. So this question is also based on an R function. Has anyone got the answer yet? So this set.seed function, it is a function in R that is related to producing random numbers. So there are these random number generators that are used, especially in the case when we want to um, set some initial conditions. For example, when we do Disney or TSNE, the we will have an initialization that is random, like for the random projections. So it's important that before running the Disney, so that if we want to get the same results for when you run the analysis, then the initial seed has to be the same, or the initial random localization that we do has to be the same. Sorry, the random initialization that we do has to be the same. So that's why we use the set.seed function to set the seed for the random number generator to achieve reproducibility. Because depending on the initial conditions that we set, the Disney results can be different. So that's why we keep the set seed function so that we get the same reproducible results when we run it many, many times. Okay. Okay. If you have any doubts, you can just raise your hand. Otherwise, I'll go to the third question. Okay. So this question is about covariance matrix in PCA. So they have said that, let me get my, yeah. So they have said that we have D variables in N samples and the data is represented as a N cross D matrix. So if it's an N cross D matrix, then um, maybe this is sample 1, sample 2. So these are the rows, and maybe this is A1, A2, and these are the measurements. Like there are D number of the um, variables. So these are the variables, and these are the samples. And they are asking what is the number of rows in the covariance matrix. So for an n cross D matrix, we have to plot the covariance between all the pairs of variables. So here the variables, like these are the D variables. These are D in number. And if we plot covariance between these, then it would be a D cross D. So the correct answer is number of rows would be option A, which is D. Because this covariance matrix is the covariance between each pair of all the D variables. So it doesn't matter if maybe n greater than D or n less than D. If you have D variables, then the covariance matrix is for the all pairs of variables. So it would be a D cross D. So it will be like A1, A2, A3, AD, and A1, A2, A3. So this will be the covariance matrix where this will have D rows and D columns. I hope it's clear to all. Anybody has any doubt, or can we move on? Okay, let's go to the next question. So once you have the answer, you can type in the chat box. It's better that you solve it during this time instead of just watching me solving it. Sorry, I moved the slide by mistake. Yeah. So just keep a note that the question is asking which of the following statement is wrong. So don't get confused. It's not asking for the correct statement. It is asking for the wrong statement. So think carefully. Yes, some of you have got the answer. So let's discuss it now. So in the question, all the options are about covariance matrix, which we just saw in the previous question as well. So let's look at the options one by one. So we saw that the covariance matrix is between all pairs of the variables. Yeah, just right. So it has, if there are n variables, then it would be a n cross 1, n cross n matrix. And these diagonal elements like x1, comma x, variance between x1 and x1, variance between x2 and x2, and similarly variance between xn and xn. These are the diagonal elements, right? So these are variance between the same variable. So it's like, or so it's not exact, it's not a covariance because it's for x1 and x1, x2 and x2. These are for the same variables. So diagonal elements are just the variances of all the variables. So option A is correct. And option B, covariance matrix, is a symmetric matrix. Option B is also correct because if you look at the terms, this is x1 and xn, similarly x1 and xn. So I hope you all know the definition of a symmetric matrix is that X transpose is equal to X. So for example, here, this term for X1 and Xn, that is the first column, nth row. First column, nth row, basically this term is the covariance between X1 and Xn, which is the same as the term here. This is also covariance between X1 and Xn, and this term is also covariance between X1 and X1. This is essentially the nth column, first row. And this will be true for all the terms. Like if you have x2, then this term would be covariance of x1 and x2. Like that would be first column, second row, and second column, first row would be the same. Similarly, third column, first row, and first column, third row would be the same. So it's a symmetric matrix. So option B is also correct. And then we have option C. Option C is all eigenvectors of a covariance matrix are orthogonal. So this you can say from the previous statement that it is a symmetric matrix. So the eigenvectors are orthogonal for a symmetric matrix, that we have seen it from the previous sessions itself. And option D, a covariance matrix is a rectangular matrix. This is not correct because covariance matrix, it's a square matrix. It's not a rectangular matrix. So the wrong answer, the wrong statement is option D. So the correct answer is option D here. Okay. Any doubts or can we move ahead? Okay, I hope it's clear then. Let's go to the fifth question. Okay, let's discuss the answer now. The question is about a single cell RNA-seq experiment. They have checked the expression of 12,000 genes in 40,000 cells. So you can imagine how much big data set this is. And each of these cells is considered as a data point. So what this means is that each um, data point has to represent all of the 12,000 genes. And they're asking what is the dimension of the gene expression space in this experiment. So they're asking dimension of gene expression space means you have to check how many genes they have collected from the experiment, and it is 12,000 genes. So the dimension of gene expression space will be just the number of genes, which is 12,000. Okay, if there are no doubts, we can go to the next question. Okay, let's go to the next question then. I hope this is clear to all. So in the previous question, I just saw the comment. They are asking what is the dimensionality. They have specifically asked for the gene expression space. So you just have to consider the number of genes, which is 12,000. So that's why they have specifically asked it for the gene expression space. So it is the number of genes. And let's discuss the sixth question now. The sixth question is which of the following algorithm is recommended to be performed on Disney on data with a very large dimension? So um, suppose the data set it's very large, then it's difficult to perform Disney. So first we can use a different dimensionality reduction technique like PCA. And based on the PCA that we do, and then based on that, we can do the Disney analysis. So if this is useful when the data set is too large, so you first, as a first step, you can reduce some dimension with PCA, and then you can do a Disney. So this will reduce the noise and this will also reduce the time taken. This is the next question. Has anybody got the answer yet? I'll give you 1 more minute, then we can discuss the solution. So they're asking in PCA, we project the data on the eigenvectors of the covariance matrix, and which of the following holds the projected data? So let me get my pen for this. So we can also look at the options one by one. The first option is covariance matrix. Yeah. And this covariance matrix we saw, it's the covariance between all the variables like xn and xk. So this is not the correct answer. And option B is the loading matrix. So this loading matrix, uh, it has information about the eigenvectors. So the eigenvectors are nothing but the principal components. So suppose your initial data looks something like this. And then suppose this is one of your, I'm just drawing one principal. Okay, I'm on the previous slide. Yeah. Um, and maybe after doing the covariance matrix and all, you have found the principal component. This is one of the principal component to be this. So the information about this principal component line or this eigenvector will be contained in the loading matrix. So these are the steps. You have to find the covariance matrix, and then you have to find the eigenvectors, which will be in the loading matrix. And then the next step is you have to project your points to the eigenvectors or the principal component. So these would be the x would be the projections, right? And this projected uh, data points, these are stored within the scores matrix. So different color. So these are the projected data points, and these are stored in the scores matrix. So that's what they have asked. Which of the following holds the projected data? The correct answer is option C. It is kept or it is saved in the scores matrix. Okay. Any questions? I'll just wait for a minute. Then we can move on to the 8th question. So the 7th question, correct answer is option C. And okay, we have 7 questions today. Okay, so we have discussed the revision of the session and then some problem solving as well we have done. So now let's go to the R session. So I hope all of you have already downloaded the codes. So we have codes for the 3rd lecture of week 8 and 5th lecture of week 8. So these codes are available in your Swayam portal itself. If anybody doesn't have the code, you can just raise your hand. I'll copy paste the code and you can run it. So all of you, we are doing the coding session now. So please open R. First, we are looking at the lecture 3 week 8 code. So let me share my R screen now. Studio. I hope it's visible. So this coding is for finding the principal, doing the principal component analysis. So first we have to read the data using read.csv. This function you must be well aware by now because we have used it many, many times. So before we go to the next step, let's just see what is in the data so that it's easier to work with. Data. Sorry. So this has some information like there are different groups, A and B, and for each group, so they are looking at 6 different genes under the condition of 3 different drugs. And what is this line doing? So this line is taking just the third to the fifth column. So we can check once if you want. If you're not sure what this line is doing, you can just print and see. You can use a print statement to see what it has done. So it has taken the third to fifth column separately. So this syntax means that it has taken all the rows. So that's why the before the comma there is nothing. It has taken all the rows, and after the comma it represents the columns. Columns are column 3 to column 5. So it is 3, 4, and fifth columns. And now the next step is to perform PCA. So to perform PCA, we use this PRCOMP function. And you know that we perform PCA just for the different variables. So here are 3 drugs or the different variables. So we have to perform PCA only for these 3 drugs. So that's why the PCA is performed only for the drugs data, which we have separately extracted here. So now the PCA is done. And these are the outputs of PCA. So it has made it into, so now we have 3 principal components. And for each component, we get a proportion of variance and cumulative proportion, which is indicative of which of the components has more variation. And this, to visualize this, we can plot something called a scree plot. So this scree plot, what it will do, it will plot the variance of each principal component. So first we are calculating the variance of each principal component so that then we can get the proportion of variance, which is saved in this variable called PV. So now we have got the proportion of variance. The plot is here. So as you can see, the principal component 1 is having the highest proportion of variance. And in the next line, so this was just the proportion of variance. We can also plot it cumulatively. So to plot it cumulatively, we use a cumulative sum function of the same variances that are here. You can also check what this data is using a print statement, just so that you get an idea. And cumulative just means that it will keep adding up with the previous term. So in PV, it was 0.6, 0.376, so on. But if we print the cumulative one, you will see that it has been added up. So, sorry, CPV. So if you're not sure about what a function is doing, you can just type a print statement after that to see what is the output. Then you can get an idea. So the first term is the same for PV and CPV, both are 0.605. But for to get the second term for cumulative sum, we have to add the first term and second term. And to get the third term, we have to add the first, second, and third term. So when you add all the three terms, because there are just three principal components, the total sum of the variances have to be 1, which is what we get here. So that's why in the plot also you can see that for the principal component 3, the cumulative variance is 1. Okay. So now we have plotted it. So now let's have a look at the loading matrix. So this loading matrix we discussed previously that this has information about the each of the principal components or the eigenvectors of the covariance matrix. So how to get this? So this loading matrix we are saving in a variable called d.load, and to get this from the PCA, this we this is the PCA that we did, right? So if you look at the output here, let me show you. So we get the three principal components. And then essentially we rotate these principal components to make them the prop X and Y axis of the plots. And the next thing is to find the scoring matrix. So this scoring matrix is the projection of the data points on the PCs. So I think this one we have to install first. So we are using a new library called ggbiplot. So typically we have been using just ggplot. So you have to install it. So it's mentioned in the starting of the code itself. So I'm just uncommenting this line, or I can also just install it from here. So for me, it is still loading. Let's see, it's still downloading because it's a new package. Once it's downloaded, we can finish the rest of the. Still, yeah, okay, it's done. So now I've finished. Uh, so you can also do the same. You can just copy paste this and run it on the console. These two lines. Okay. I'll just wait for 1 minute so that all of you have downloaded this. First, run this line and then just copy paste it here in the console. So the console is in the bottom of your screen over here. I hope you can see my cursor. And then it will take 1 to 2 minutes to download the package. Okay, so I hope we can go to move on. Okay, so now let's use the new package that we have just downloaded. Oh yeah, I didn't call the library. Okay, so I've called the library. Now let me run it. And let's print it. Yes, so you can see that these are the Let me just make it bigger. Yeah. So this is the output of the plot of the three principal components. So you have PC1, which explains about 60%, 60.5% of the variance. And then PC2 about 37.6 on the perpendicular to the PC1. And based on these, we see the gene 1, gene 2, G3, and so on. So now some of the data we can't see. The plot is from, if you can see, it's from -1 to 1, both x-axis and y-axis. So now we are setting both x-axis and y-axis from -2 to +2 so that the data is better. And finally, we can color code them based on the groups as well. So now it has been divided into two different groups based on the PCA analysis. So these were the two groups, A and B, that we saw in the input data itself. So this was about A code. There is one more. There are two codes, two scripts for lecture 3, week 8. So I'm opening the second B script. You can all open it as well. So let me just clear everything here. Okay. So here we are going to work with gene expression data of 64 cancer cell lines. And we are looking at a total of 6,830 genes. So it's, you can imagine it's a 64 cross 6830 matrix. So we are first reading the data. Um, so you can see what the data is by print statement. Should be the same that we read in the description, that it's the, so these are all the different genes. So as you can see, it's a huge data set. 6,830, right? So if you keep scrolling down, yeah, we have up to 6,830. And so you should remember that this 6,000, these values are for one particular cell line, but in the data set we have 64 cell lines. And the data, it's not able to print everything because the data set is so huge, okay? So now we are getting the parts of the data that we need for the PCA. So we have taken just the last. So -1 means last column from the last. So it's the last column. So now we have done the PCA. Now let's get the scree plot to see which of the components are important. So the scree plot, like typically it shows a decrease with more higher principal components have lower scree plots. Sorry, lower variance explained. But if you do the cumulative plot, then we can expect that the trend will be opposite, because we saw that for a cumulative plot, we keep adding the numbers, adding the previous numbers. So obviously the numbers will get bigger and bigger. So if you run this code, then we will see that as the principal component increases, the Y value, or the cumulative PV value, actually increases. And now we can again plot it with the principal component that we got using this ggbiplot. So here the principal component 1 is explaining only about 11% of the variance. So this you can see from this plot here as well, that this value is about 0.10. But in the previous example, if you remember, it explained about 60.5. But here there are too many principal components and too many genes. So that's why the principal component 1 is only accounting for 11% of the variance. And principal component 2 is about 6.8% of the explained variance. Hmm. And now we can also, for each color, like each dot here represents the cancer cell lines. And we can color code these points based on what is the cell line. So I hope this was clear to all. So these were the two codes from the third lecture of week 8. If you are done with this, we can go to the lecture 5, week 8 lecture code. So here we just have one script to run. So I'll wait for 1 or 2 minutes for you to open the script, then we can run it. Meanwhile, I'll just delete all my history. Yeah. So here we are going to look at dimension reduction using TSNE, or T-distributed stochastic neighbor embedding, using single cell gene expression data, which is a published data set. Okay, so I have not. So again, here we are using a new library. So first we have to install it. Sorry. So I'm installing it from the console using. So I'm using the install.packages in R. So I think it must be downloading now. Let me check. Oh, it says it's not found. Let me try again. Sorry. Now it should work. Yeah, now it's downloading. Okay, so this is done. ggplot also I've already installed it. Like we have been using it in the previous classes as well. So now let's read the data set. So here, as you can see, this is the data set, some single cell gene expression data. Meta. Open the connection rather than fail. Okay, I'm not able to open it. So I'll try giving it as a path. Yes, path name. This is const.csv. Okay. Yeah. So after I gave it as a path, it is working. I have them in the same folder. I think it's okay. What's my working directory? So typically, if you're in the same, if the files are in the same working directory as um oh, okay, sorry. Yeah. So it's in the third lecture, but now these files are in this lecture 5 folder, but the working directory is in lecture 3 folder. That's why I'm not able to load these files with just the file name. So if the working directory and the folder or the file path is different, then we have to give the entire file path. Or if you want, you instead of that, you can also change the working directory. Yeah. So yeah, now it has read the data. Oh, sorry, I just saw your message, Sudha. Um, the R codes are available in the Swayam portal. So where you can download the slides. So in some of the classes where there are R codes, you can download a zip folder. And if you unzip the folder, from there you can get the R codes. Anyways, I'm just copy pasting the code for you. So for now, you can just copy paste the code from the chat box. But the data set, you will have to get it online. Let me check if I can put it in the chat box. Um, uh, Meta. I'm not able to put the data sets in the chat box. So you'll have to get it from the PIAM portal itself. If you have any doubts, I can maybe show you also how to get it. So if you're not able to get the codes, just raise your hand. I will show you from where you can get the codes. Okay. Okay. So let's continue. Where were we? Yeah. So we were here. We have finished reading the data. So typically we have something like in many biological data sets, we have something called as metadata. So this metadata is nothing but it has information about the data, especially for large data. So this has the cell type information is contained in the metadata, and we have 4 different cell types, like 4 different neural cell types. So this you can get the name of the cell types, you can get it is present in the metadata within the column inferred.cell.type. So let's get that. And now for Disney, we saw that first we have to set a seed of random number generator so that it can be reproduced. So this we saw in the example questions. We have now this is done. And now these are some of the important arguments uh, for the R-Disney plot. So we have the important arguments are perplexity, and then we have the dimensions or the output dimensionality. So by default, it is 2. And then we have PCA. So this PCA, if you want, you can um, do it when the Disney data plot is already big. Um, and then the next is check duplicate. So especially in large data frame, maybe there are some data points that are duplicated. So that by default is also true. And then we have the ETA, which is nothing but the learning rate. So typically, the suggested range is from 10 to 100. And then we also have the theta. So that is like a speed accuracy trade-off. So if you, so that will also depend on the rate. So if the code runs faster, then there may be problems with the accuracy. But if you want good accuracy, then the time taken to run the code will be faster. So let's also, this perplexity is a measure of how many number of nearest neighbors it considers. So that is the perplexity, number of nearest neighbors. And then the second term is straightforward. So this is an important term that will determine the quality of your Disney. This term is straightforward. It's the output dimensionality. So typically it is 2, but for visualization, 2 would be the easiest. But if you think that your data is extremely complex and maybe 2 dimensions may not represent your data completely, then you can go for 3 as well. And the default value is 2. So if you want to set it as 3, then you have to set this argument separately. So here, if you can see perplexity, we have set. So this DIMS, this argument we have not set. So by default, it would be 2. And PCA also, whether we have to perform PCA or not. So this is also straightforward. And the next argument, check duplicates, is straightforward. Um, and then we have ETA or learning rate. So this argument controls how the embeddings adjust at each iteration. And then finally, we have the theta value, which is the speed accuracy trade-off parameter. So if it is 0, then it is exact Disney. So exact Disney means it runs very slowly, but it is accurate. And finally, we have the max iter or the number of iterations. So the more the number of iterations, the better would be the convergence. But however, if you increase this number too much, then the computational time will also increase. So now let me run it and check how much time it takes. So as you can see, the code is still running. It's so because in this there are many iterations that have to be performed. So it is a computationally expensive process and it's taking some time to run here. So we just have to wait till the code finishes running. Meanwhile, does anybody have any doubts? Okay, so it is done running. So you can see it here. So initially that stop symbol was there. And you can also see it here. It has moved on in the console. It has moved on to the next line. So now we have to visualize the output. So for that, let's first get the results as a data frame that can be plotted. And we can plot it using ggplot. So here we have 2 axes because uh, the default number of axes is 2 and we didn't set it here. And here we have set ETA, perplexity. So you can try to play around with these values and see what happens. I'll just run the entire script. Then we can change these values and see how the results change. Actually, we can try it now also. So right now the Disney plot looks like this, where all the interneurons or the orange points, you can see they seem to be forming a cluster. And all the neurons which are in blue color, they are forming the cluster. So there is The output seems to be making sense. So now what we can try is we can try to, mm, so let's try to run it faster now. So the exact default theta is 0.5, but if we give a different value, say I'm setting it to 0.9, now this will run much faster than before, but it will lose the accuracy. So here we are seeing, in this case, we are seeing the clusters have been separated based on the cell type. Let's see what kind of an output we get when we set the theta to 0.9 and decrease the computational time, and the accuracy will decrease. So even then you can see it's still running. We can also reduce the number of iterations to make the code to run faster. So now if you plot it, so now let's plot it again and see how the plot has changed. See, now the plot has become very different from before. But obviously the plot that we got first is the better plot, because there the theta value or the trade-off value was lesser, meaning that accuracy was preferred. But with theta is equal to 0.9, speed is preferred, so we are compromising on the accuracy. So let's stick to the default theta is equal to 0.5. And yeah, the code is running again. So I hope all of you are with me and you're also getting the same kind of different plots with when we play around with these Disney arguments. Okay, so I guess it has finished running. Yeah, it's finished running. So now we get it as a data frame and then we have to plot it. Okay. So here each dot represents a particular cell. So a violet dot is an RG cell, and there are many such RG cells. That way you see many plots. So this is for all of the genes. Now suppose we want to just look at one of the genes. So then you can specify that. So here each dot, again it is, each dot is again for each cell, but these dots are also color coded, meaning that gray color dots means that particular cell is having a lower gene expression of this neuro D6 gene. And red color means it is having a higher gene expression. So for example, if you look at the top left cluster of points, they are all having lower neuro D6, and these are the cells correspond to the RG cell type. So you can infer from these two plots that the RG cell type is having lower neuro D6. So you can compare these two plots. So this lower left is having higher neuro D6. Let's see which. So these mostly correspond to the neuron cell type. Okay. So that's it for today. We have discussed the concepts, we have discussed some problems, and we have also run some R script. Um, if you have no doubts, then we can end the session. If you have any questions, we can take them and then we can end the session. Does anybody have any questions? If you don't have any doubts, you can leave the meeting. We are done with the class. If anybody has any doubts, we can take questions now.
Mm. Pretty Casey. Who's this? Hello? Hello?